Hello, everyone. So today we're going to talk about biophysical properties of lipids. So before we jump into this and we use what we learned about the biophysical properties of lipid to understand how we can use this to separate and identify lipids in a sample, I want to talk a little bit more about polyunsaturated fatty acids. So considering the real life biochemistry, the questions that I brought for today are, have you ever thought why levels of omega-3 and 6 in our diet can affect our health? Have you ever thought why bears can hibernate standing extreme cold weather? So as you go through the content, we are going to be able to answer these questions and understand a little bit more about this process. So some cis polyunsaturated fatty acids we cannot synthesize, and this is the case for an omega-3 fatty acid that is called alpha-linolenate, that is also called as ALA, and it can be found on vegetable oils. Another examples of omega-3 fatty acids are eicosapentaenoate, that is the EPA, and docosahexaenoate, that is the DHA, and these are found on fatty fish. What is interesting about the polyunsaturated fatty acids is their nomenclature can be a little bit different. So last class we talked about that we call carbon number 1, the one that is a part of the carboxyl group, carboxylic group. And in the case of polyunsaturated fatty acids, since their physiological role is more related to the position of the first double bond near to the methyl end, right here, we call this carbon number 1. It's the carbon that we called omega. So we start counting here carbon 1, 2, and 3. And so in this case, the first double bond is on carbon 3. So that's why this fatty acid here, it's called an omega-3. Here we have more examples of omega-3 fatty acids. As I said, the ALA, we have the EPA and the DHA. They all have the first double bond on carbon 3, starting to counting from the methyl end. And here examples of omega-6 fatty acids, where the first double bond, it happens between carbon 6 and 7. Remembering that we have to start to count from the methyl end right here, okay? And examples of omega-6 fatty acids are the linoleic acid, LA, and the arachidonic acid that we can also call as AA. So here you can see the pathway for the synthesis of polyunsaturated fatty acids, omega-6 and omega-3. And it's important to note that mammalians cannot synthesize the omega-3 ALA, but they can synthesize EPA and DHA from the ALA. The problem is the amount that is synthesized, it's not sufficient. So it's very low. So it still depends on the sources from the diet to have proper amount of these polyunsaturated fatty acids. I want to call your attention here to the importance of fatty acids in the scientific field. So I just wrote omega-3 fatty acids in the platform on the website of PubMed, where we have the scientific publications available. And I selected the published article, including omega-3 fatty acids from the year 2020-2021. So what I got was 1,760 published papers on this topic. And these are related to many issues like ADHD, autism, rheumatoid arthritis, cardiovascular disease, chronic obstructive pulmonary disease, COVID, cancer. So this is just for you to get a sense of the importance of the omega-3 fatty acids in the scientific field right now. So considering polyunsaturated fatty acids, omega-6 and omega-3, what is most important is the ratio that we have between these two. So Studies say that omega-3, EPA, DHA, they have anti-inflammatory properties. On the other side, omega-6, as arachidonic acid, they have pro-inflammatory properties. So in our current Western diet, we have a ratio of 25 omega-6 for 1 omega-3. And this seems to be related to a higher incidence of cardiovascular disease. And for example, on the other side, when we consider like the Mediterranean diet, when we have an increased content of fish and nuts, for example, and vegetables, the ratio the ratio between omega-6 and omega-3 is much lower. So what is recommended for a healthy diet, it's a ratio between omega-6 to omega-3 of 4:1. So what is the mechanism that has been behind the actions of the omega-3 properties? And studies have shown that polyunsaturated fatty acids, they are able to inhibit the HPA axis, that is the axis that is responsible for the pathological stress response, and for the stress response actually. And then when you have the action of the polyunsaturated fatty acids, they inhibit this axis, and this leads to the reduction of pathological stress response. So for those who are not familiar, the HPA is the hypothalamic pituitary adrenal axis, but there are also another types of actions that the polyunsaturated fatty acids can exert, and one of them is through the production of stimulation, the production of BDNF, and this inhibits cellular damage. And on the other side, can also stimulate the production of resolvin and neuroprotectin that inhibit inflammation, and in this case, reducing cellular damage, and also inhibiting the production of reactive species, oxygen species. So avoiding oxidative stress and cellular damage as well. So we talked about different types of fatty acids. We have saturated fatty acids, unsaturated fatty acids, polyunsaturated fatty acids, but how do we store these fatty acids? And the way you do that is through triacylglycerol. And the major site of accumulation of triacylglycerols is the adipose tissue. When we have these adipose cells full of triacylglycerols, and their nucleus are dislocated to the periphery. But why don't we store just fatty acids? Why do we need to get the fatty acids built to triacylglycerol and then store them? And the answer for that, we already talked about it, and it's because we know that in the physiological pH, the fatty acids, they are in the ionized form. So if I have high concentrations of free fatty acids in the cells or in the body, this would disrupt the pH balance, and that would be a problem. So that's why we store them as triacylglycerol. And just to show you here that the triacylglycerol actually, we have a glycerol backbone that is connected to the fatty acid chains through an ester linkage. But why do we use triacylglycerols as stored fuels instead of polysaccharides like glycogen, for example? Wouldn't it be much easier? We have glucose, we can make glycogen, and then from the glycogen, we can make glucose and use glucose as a fuel. We need another source. So the problem is that the carbon atoms of fatty acids are actually, the answer for this is that the carbon atoms of fatty acids are more reduced than those of sugar. So in this case, they are able to generate more energy. And the other reason is, if you think about the triacylglycerols, they are hydrophobic. So in this case, you don't have the extra weight of the water of hydration that happens with glycogen. So that's another reason. And when we have the triacylglycerols, if you look at these two examples, we can have exactly the same fatty acids in the composition, three same fatty acids in the composition of the triacylglycerol. And in this case, we call it a simple triacylglycerol. On the other hand, when we have different fatty acids in the composition of the triacylglycerol, then we call it a mixed triacylglycerol. So triacylglycerols, they have this function of energy storage. They are stored as fat droplets in cells. They have the property of giving the insulation against low temperatures. That's important for seals, walruses, and penguins that live in low temperature environment. They are also important for hibernation, when we have huge fat reserves accumulated before the animals go into the hibernation. And this is important for insulation and also for energy storage. So here's an example of a polar bear, but for us to be more interesting, you think about grizzly bears. So you know that during the summer, the grizzly bears and the black bears are building their storage of triacylglycerols to be able to hibernate. And again, it's not just the energy that they're looking for. It's also the property of thermal insulator that is going to help them to maintain the body temperature. What if I need the fatty acids? How do they circulate? I'm going to degrade them from metabolize them from triacylglycerols. I'm going to get the fatty acids, and then how do they circulate? So fatty acids, free fatty acids circulate in the blood bound to non-covalently to albumin, as you can see here. And the fatty acids are present in blood plasma mostly as carboxylic acid, derivative such as derivatives, such as esters or amides. So now let's move on to how we identify the lipids, okay? And so as I said, we can use their physical property and and their solubility and the way they behave to separate this in a sample. So actually we have that the separation is based on differences in polarity or solubility in non-polar solvents. So what happens is that lipids that contain esters or amide are linked fatty acids. They can be hydrolyzed by treatment with acid or alkali, or even with specific hydrolytic enzymes as phospholipases, glycosidases, to yield components for analysis. So what all that means, we're going to see in the sequence. So this is a scheme of how we identify lipids. So we have a group of cells, or we have a tissue. The first thing we need to do is homogenize this tissue using solvents. So here there is a mix of chloroform, methanol, and water. And when we add our sample to this mix of solvent, what is going to happen is, depending on the property of our lipids, some are going to remain in the methanol water mix, and some are going to move to the chloroform mix. And based on these properties, we are going to be able to further separate them and identify. So here we have two approaches. One, it's called the shotgun approach, that is direct mass spectrometry of the total extract. That is a faster approach to identify these lipids. And the other option is that we can separate these lipids then using thin layer chromatography or adsorption chromatography, gas chromatography, and HPLC. And then we do the mass spectrophotometry of different types, conditions, and we get to the lipidome, okay? So let's talk a little bit about each step. So the extraction, what is going to happen, as I said, depending on the characteristic of the lipids that we have in the sample, they're going to be distributed through the methanol water mix or to the chloroform. So neutral lipids like triacylglycerol, they are soluble in ethyl ester chloroform or benzene, so they're going to be in this phase. On the other side, membrane lipids, they are going to stay in the phase that has ethanol and methanol. So this is the first step to separate these lipids and start to being able to identify them. So after that, as I said, we could go to the shotgun or we could go to the thin layer chromatography. And the approach that we use for the thin layer chromatography is that the less polar lipids, they travel farther than more polar or charged lipids. So you can use different dyes to identify these different lipids, and then you do the mass spectrometry. The other approach that we can have is the absorption chromatography that is based on different polarity. So here we are going to have our sample. It's going to be homogenized in the solvent, and then it's going to separate. Some lipids are going to stay in the methanol water, some in the chloroform, and then we are going to do the absorption chromatography. And as we increase and as we add different solvents, we are going to be able to separate it, to separate different lipids based on the different polarity. So, for example, polar lipids, they are going to bind to the polar silicic acid. Neutral lipids, they are going to pass directly. When we do the first wash with chloroform, polar lipids, they are going to be eluted by increasing polarity. Uncharged but polar lipids are going to come out when we use acetone. Very polar or charged lipids are going to be eluted with methanol. So use different solvents with different, we are going to be able to separate the lipids of different polarity. We can also use gas chromatography that separate volatile components. So tendency is to dissolve either inert material in the chromatography column or to volatilize to move through the column in an inert gas such as helium. So there will be the separation based on this property that the specific lipid we have in the sample have, if it's capable to be volatilized or come out with the gas, or if it's going to be dissolving the column. And some lipids are naturally volatile, but others not. So an approach that can be used is to increase the volatility. And we do this using the heat. So we heat sample in a methanol fluoridic acid or methanol hydroxysodium to convert acids esterified to glycerol into their methyl esters. So the gas chromatography, the ferrosyl esters that are most soluble in the column material dissolve in that material, and the less soluble lipids, they're going to be carried by the stream of the inert gas, as I said before, and they're going to emerge first from the column. So the order of dilution in this case is going to depend on the nature of the solid absorbent of the column and the boiling point of the component of the lipid mixture. So as I said, there is a more direct method that is the shotgun, where we do the mass spectrometry. And then in this case, the components of the lipid mixture can be separated and identified by their unique pattern of fragmentation. We have here high resolution that avoids losses during the preliminary separation, and it's also faster. So that's the advantage of this approach. There are also other strategies that can be used, as I briefly mentioned at the beginning of this talk. With mild acid or alkaline treatment, we can have acid-linked fatty acids, triacylglycerol, phospholipid, and esterol esters that are released. We can harsher hydrolysis and release amide-bound fatty acids from sphingolipids. So we can use different strategies to be able to separate the lipids that have different characteristics. And we can also use enzymes that specifically hydrolyze certain lipids. So this is an example of a lipid that is called phosphatidylcholine. That's a component of the membranes. And we know that there is this enzyme that is called phospholipase C that is responsible for catalyze the reaction when we have the production of phosphocholine that is water soluble and diacylglycerol that is chloroform soluble. So we can use this specific enzyme that is going to be able to cleavage in this specific position and obtain these subproducts that we can then identify. So to finalize, it's important to understand some concepts related to lipids so you can better understand the process of identification. So in lipidomics, we call lipidome the full complement of lipid-containing molecules in a cell, organ, or tissue under a particular set of conditions. And lipidomics is the systematic characterizations of the lipidome. There is also what we call lipid maps, and through this you can see specific fat acids that are designed by numbers. So each individual lipid receives a 12-character identifier that is the LMID of this specific lipid. Okay. So here we have our learning objectives, and I'm looking forward to see you in class so we can learn together and discuss a little bit more about these topics.